Okay, good morning to all the participants. Today is the last day of our week 2 module introduction to bioinformatics. And today we'll talk about drug development, drug discovery, and topics like computer-aided drug designing, including molecular modeling and docking processes. Okay, so I'll start the session, today's session, with what is a drug, what is the process of drug discovery, and what do you understand by the process of drug development. Okay, what are the steps involved and the drug receptor theories. All this will be covered today in my session. Okay, so I'll just start. Okay, so this is drug discovery and development, today's first session for the day. And here you can see a small chart. Okay, so this is a small picture representing the drug development pipeline. Okay, so if you're aware about what is a drug, then drug is supposed to be medicine in the layman language. What the common people call it is something which we are using in order to go for the treatment of any disease. Okay, so this is a small pipeline. You can just understand through this pipeline that in the process of drug development, we will cover actually these things. We need to understand how the drug is actually discovered, how the drug goes for the preclinical trial phases, what about the clinical trial phases, and how the drug is gone for the approval. So basically, when we want to produce a drug, okay, so going with the development of a drug, we need to follow all these steps. The discovery of the drug is necessary. The drug should go for preclinical and clinical trial phases before the approval. Then only we can just go with the drug in the market and it can be consumed by the patients. Okay, so moving ahead with the slide, here the process is represented in a detailed manner, because I told you that drug is something that is supposed to be a kind of medicine in the common people language and used for the treatment of disease. So this actually undergoes these several phases. Okay, so whenever we are consuming a drug, we need to understand that this drug will be going to get binded at the site of action. Okay, so drug will be consumed by us. It will be consumed orally or intravenous, and there are other mediums as well through which we can just consume the drug molecule. Okay, drug molecule as in the medicine. So we are consuming this, and the moment it will get inside the body, it will be absorbed, distributed, okay, and it will reach its site of action. So the first thing is we need to understand what is that target, what is that receptor site where our drug will bind. Okay, so this drug discovery process includes the first thing, that is we need to select a target, a place where drug will go and bind. And when we are going to study the process of target selection, then we go with the study of both cellular and genetic targets. Okay, we need to understand what about the change in the genome of the cell. Is there any kind of change in the genome or any kind of change? Is it being involved in any other changes in the genetic material or the production of proteins? Okay, and the involvement of bioinformatics is obviously necessary, because whatever process we'll be doing, or we'll be going with the study of development of a drug, a phase comes that is said to be computer aided drug designing. So in order to search for these targets, for these receptors, we can use bioinformatics as a branch to go for virtual study. Okay, the study of how the drug interacts with the target site. Besides this, we need to understand that we need to select a certain compound that will be supposed to be a lead compound, and we are considering that this lead compound is the best of all, and this will work for our disease, for the treatment of our disease. Okay, so one of the phases of drug discovery process is we need to go for our lead selection, lead discovery. Okay, how to synthesize it, how to isolate it. Okay, combinatorial chemistry will help us to go with the study of the chemical structure of our lead compound. We need to develop an assay if we want to go with the treatment of the disease while developing a drug. We need to go for an assay. But what kind of an assay we will apply, what will be the procedure to go with the process of in vitro study or in vivo study. And obviously, when we are selecting the lead compounds, we are selecting it from a library of compounds. So high throughput screening is necessary. We will select the compound, a lead compound, from a big database from a chemical library. Okay, and medicinal chemistry, obviously, whenever we are going with the study of any drug molecule which is supposed to be a chemical compound or any compound that is being extracted from the natural source, okay, so we need to undergo the study of its structure. And if you're going with the study of the structure, we need to understand if the study of structure is there, then we need to understand how the structure is leading to the activity. Okay, so how the structure of that compound that is behaving as a drug molecule, or if you're talking about the structure of that target or receptor, so we have to go through the structure analysis of both these compounds, the receptor site molecule as well as the lead compound molecule or the ligand or the drug molecule, and study their relationship as well. So in silico screening will help us the study where we are using various web-based tools and softwares to go with the study of this kind of interaction. Okay, and similarly, chemical synthesis, the process where we are considering how the atoms are behaving, what kind of properties we need to consider to just convert our lead compound molecule into a probable drug molecule. Okay, so going ahead with the process of drug discovery, in vitro study is necessary as well as in vivo study is necessary. Okay, going through the in vitro study, we will come across the study of various properties talking about affinity, efficacy, okay, and what about the mechanism of action, how the lead candidate molecule is working, is it effective or not, okay, and the similar study will go with the animal models in the in vivo studies. Okay, so all this will be there in the process of drug discovery. And finally, going with the preclinical trials, animal testing, we need to go for clinical trials again. Okay, because we need to understand how these drug molecules they are behaving on human body. So we will take patients, we will see the effect of the drug on the patients as well as healthy volunteers. And all this, after going all these steps, then the drug is supposed to go for the approval by the drug federation, where the drug is being approved to be there in the market, okay, to be available in the market. And if there are no side effects, then the drug is being manufactured and it is supposed to be consumed by the patients who are suffering from that particular disease. Okay, so this is a very brief summary about drug discovery. What is that? We'll just start with the presentation. The first thing is, so the term drug discovery is actually referring to a process where we are considering how a new candidate drug is discovered. Okay, so whenever we are going with the term called drug discovery, we are discovering a drug that is a new drug, a novel drug. We never go for a process of drug discovery and development for any drug that is already available in the market, which is already being prepared by the pharmaceutical companies. Okay, so it should always be a novel, a new candidate drug. So previously, historically, what was being done is the drugs, they were actually being discovered from the active ingredients, which they were just extracting from all those traditional remedies which they were using for certain diseases. Okay, so previously the drugs were extracted as the compounds or the active ingredients from the traditional remedies, or by serendipity, by accidental discoveries also drugs were being discovered. Later on, the classical pharmacology came into existence. Okay, and under the classical pharmacology, small molecules were screened from the cells. Okay, and their effects were being seen. Is there any kind of therapeutic effect? Is it helpful for the treatment of the disease? Okay, here the term pharmacology refers to the study of drugs. Okay, so historically what happened? First, they were trying to extract compounds which were available there from the natural sources or by any kind of accidental discovery. Then the classical pharmacology, they were trying to study how small molecules, they were screening it from different cells and organisms, and they were trying to study what are the effects on the disease treatment. Then later on, the process of reverse pharmacology came, where the targets were hypothesized. Targets are those sites where we are considering the drug will go and bind and help in the treatment of the disease. So these targets, they were hypothetically created, and they were supposing if the drug go and bind there, so what kind of action the drug will show. Okay, and presently the modern drug discovery process, it is actually being involved in the identification of screening hits, how the drug molecule is going and binding there, what about the medicinal chemistry, what about the chemical structure of these compounds, and how we can go with the optimization of these hits. Because we want to increase the affinity. Affinity is we want to go with the increase in how the drug can bind and go with the action. Okay, selectivity, we need to understand what are the basic properties, the suitable properties a drug molecule, a ligand molecule should have just to behave as a good drug, okay, for the treatment of the disease. Affinity and efficacy, binding and activation of the drug, both are important, as well as stability of the drug is important. We need to have a drug that is stable enough. We can't afford a drug that is not stable enough and it is not reaching the site of action. Then what is the use to go for the drug development process? Okay, consuming the drug if it's not reaching the site of action. And oral bioavailability is a property where we are considering what amount of drug is actually being administered to the site of action. Okay, so once the compound is fulfilling all these properties, then we go with the process of drug development. And with the process of drug development, we need to understand that computer-aided drug designing is one of the steps for the process of drug development. Okay, so the first thing is, what is a drug? So drug is actually a molecule which we consider is helpful for the treatment of the disease. Okay. And the second thing is, if we say what is a drug, so we need to understand that drug is a chemical compound. Okay. A chemical compound is converted into a drug only when it is having all these properties. If it is safe, if it is effective, if it is stable, deliverable, available, and novel. Safe refers to if it is safe to our body. Safe as in we are consuming the drug molecule for the treatment of the disease. So this compound or the drug molecule, it should be safe to the body. It shouldn't cause any kind of toxic effect to the body. Okay, so it should always be a safe molecule. The second thing is it should be effective. Obviously, whenever we are trying to consume a drug, it should be an effective drug. Like for example, we are trying to consume two drugs. One is a drug A, the other one is drug B. If I am considering drug A is causing an effectiveness of around 80% with the dosage that is working in 2 hours, okay, and the other drug is working with the effectiveness of 60% with the dosage of just half an hour. Okay, so considering both these drugs, we will consider the drug to be the more effective drug, which is causing an effectiveness, the percentage of effectiveness in a higher percentage, as well as the deliverable time should be lower. Okay, so whenever we are considering a chemical compound to be a drug, we need to understand the drug should be effective enough to treat the disease. Okay, we can't take a drug that we are considering is good enough. It can just bind to the receptor side because the structure of the target side and a drug molecule, they are parallel, complementary to each other, and we have thought that it is having certain properties. But if it is not helping for the treatment of the disease, we can't consider it as an effective drug. Okay. The next property is a chemical compound should be a stable compound. We can't afford for any such compound that is taken up by us, taken up orally, intravenous. It is being absorbed by the cells and distributed across the body. Obviously, when the molecule is being taken up, so it is not like if I'm suffering from a disease that is in my lung or suffering from a disease in the liver and I'm taking the molecule orally, so it is just targeting that site. It is actually being distributed across my whole body. Okay, and as soon as it is reaching all the cells, it will be there participating in the process of metabolism. Okay, so whenever we are trying to go with the process that how the drug is being administered, is it is if it is reaching the site of action, we also need to consider that the drug should be a stable molecule. Okay, we can't go with any such molecule which is going, but the stability is almost zero for that. Okay, it is going and it is being converted into some other molecule, or it is going and it is breaking up into its atoms. Okay, it is not a stable molecule. Then if it is not stable, it will not go and reach the site of action and won't help us with the treatment of the disease. Okay. Next is deliverable. Obviously, it should be deliverable to the site of action. We can't afford to consider any such compound to be a drug which is coming inside our body, going across the body, and it is probably excreted out from the body. It is not reaching at the site of action. It is not delivered at the site of action. Then what is the use to consume such kind of compound? Okay, so again, this property should be considered. Availability is also important, because if I am considering any compound that is not available in the market easily, then why to go for such compounds to be a probable drug? Okay, so availability of such compounds is also necessary. I started with that whenever you want to go with the process of drug discovery or development, we should go for a new candidate drug only. It should be a novel drug, a new drug. Okay, so all these properties should be there whenever we are considering a chemical compound to be a drug molecule. And as I told you, study of drugs is said to be pharmacology. So basically there are two branches under pharmacology. There is pharmacodynamics and there is pharmacokinetics. Okay, so the moment we have consumed a drug molecule, a medicine, it is taken up by us, like for example orally. It will go inside our body through our digestive tract, and it will be distributed across the body, the whole body. Okay, the moment the drug is being distributed, absorbed and distributed, okay, so all this about the absorption, the distribution, how it is participating in the metabolism of the cells of our body. Okay, what amount of drug is being excreted out? Okay, and is the drug causing any kind of toxic effect? All this will be studied under ADMET properties. Okay, so this comes under pharmacokinetics, because the moment the drug will enter inside the body, we need to understand, we need to study how the drug is acting on the body, what the drug is doing on the body. So what drug will do? It will cause some kind of biochemical and physiological effect, okay, on your body. And what about the mechanism of the drug action? How the drug is working inside your body? What the drug is doing on the body, it is studied under pharmacodynamics. Okay, and how our body is responding towards that drug, that is studied under pharmacokinetics. As I told you, it is being absorbed, distributed, the metabolism of the cells are also being affected, the amount of your drug will be excreted out, a portion will be excreted out, the whole drug is not being delivered at the target site. Okay, and we also need to understand if the drug is causing any kind of toxic effect to your body. Okay, so whenever we are going with the study of drugs, we will go with these two kinds of studies, pharmacodynamic and kinetics. How the drug is acting on the body is dynamics, and how the body is responding towards the drug is kinetics. Okay, so these two will be included whenever we are going with the study of drug, including the above properties like it should be safe, effective, stable, deliverable, available, and novel. Okay, so then is because we are consuming a drug molecule, for example. Okay, drug molecule is consumed. There is a site where the disease is there. For example, liver is the supposed site where we are considering the disease is there. We have created a drug molecule or medicine. Okay, we have consumed it. It has moved across the body, and what we are considering, what we want is the drug should go and reach at the liver. It should bind at the receptor site, and it should start its action. So the first thing is, do the structure of drug is supposedly complementary to the site of action? Is the drug is available at the site of action? Is it okay to be binded at the site of action? So we need to understand that the structure of both drug and receptor should be studied. Okay, the receptor structure should be such as the drug will go and bind to that receptor site. Okay, and whenever the drug is being created, it is being created in a structure in a manner so that it will bind to the receptor just in the manner how key binds to the hole of the lock. So we consider drug receptor interaction to be an interaction that is like a lock and key interaction. Okay, and when we want to go for the study of these receptors or the targets, we want to study that we need a receptor at the target site where a drug molecule should easily go and bind. So what is that suitable receptor? That suitable receptor is, it is a lipid molecule or carbohydrate molecule, proteins or nucleic acids. They are there part of the cell. These biomolecules, these macromolecules, they are there and present on the cell, and they can easily act as the receptors, and we can go with the study of their structures very easily. Out of all these four, the best, which is mostly considered to be the receptor molecule, is a protein, because the study of a protein structure is very easy. Mostly we get the structure of a proteins, the three-dimensional structures, from a database protein data bank. If not from the protein data bank, we can go with the structure prediction process, homology modeling. Okay, we have gone with the tertiary structure prediction process, so we can just create the 3D structure. Okay, so the most probable receptor is our proteins. Then what about how the drug and receptor, it interacts with each other? Okay, so the drug and receptor, it interacts, okay, and it follows all these theories. The first is the receptor site theory. As I told you that the drug and target, they bind just like lock and key. We suppose that receptor is having a hole just like the lock is having a hole, and drug molecule is acting as a key and it is binding to the hole of that lock. So based on this theory, we need to understand drug and receptor will bind to each other. Okay, the receptor site theory states that the receptor should be in a manner, in a structure, so that the ligand or the drug molecule can easily go and bind to that receptor site. Okay, and all the possible interactive forces, whether they are van der Waals interactive forces, hydrogen bonding, hydrophobic interaction. Okay, so all sorts of interactive forces, they should be helpful to just help the ligand or drug molecule to bind at the receptor site. Okay, the structure should be in such a manner that they should go and bind to each other and create a conformation that is a stable conformation or minimum energy conformation. The moment the drug will go and bind to the receptor site, it will be activated, and the sooner it will be activated, it will start its action. Okay, so binding and activation, affinity and efficacy is very much important if we are talking about the administration of a drug molecule. Okay, so membrane potential theory states that, obviously I talked about that drug is moving across the body, and the body is having number of organs, number of tissues, and it is made up of cells. And if it is a cell, so cell is having a membrane. Okay, so the potential of the membrane should be in such a manner that these molecules should move across the cell membrane easily, move from one cell to the other cell. Okay, so the membrane potential should be maintained in such a manner that the drug molecule should move across the cell membrane, reaching its site of action. Okay, so this is the membrane potential theory. Next comes the occupancy theory. Okay, the structure of our molecule, the drug molecule, should be in such a manner that it should occupy almost all the surface of a receptor site. We should not create any such structure that is not occupying the 100% surface area, because the more the coverage of the occupancy, the surface is being occupied by a drug molecule, the more will be its action. Okay, the sooner will be its action, the response will be in a higher speed as compared to a drug molecule which is not occupying the whole of the surface area. Okay, rate theory is also important. Why rate theory is important? Because rate theory is helping you to understand what about that time duration in which we are considering the drug is being binded at the receptor site and showing its action. Like I've given you an example. For example, I'm taking two drugs. One drug is A drug, the other one is B drug. A drug is leading to an efficiency. It is having an efficiency of 60%. Okay, the drug is efficient to around 60%, and the 60% efficiency of the drug is, it is taking a duration of 4 hours. So one dosage is helpful. It is 60% efficient. Okay, efficient as in it is being distributed across the body, it is reaching to the site of action, it is binding to the receptor site and starting its action. Okay, for the treatment of the disease. Okay, so 60% efficiency is there and taking a time period of just 4 hours. On the other hand, if I'm having a drug B and the drug is helping me to just go with the treatment of the disease, and that is around 85%. But on the other hand, this particular drug, the time it is taking is a time period of 12 hours. Okay, so comparatively, if I'm comparing the rate of action, if I compare A drug and B drug, my A drug is just going with a time duration of 4 hours and the B drug is taking a time of 12 hours. So when I'm taking one dosage of B drug, that is for 12 hours, the other drug can be taken in 3 dosage. Okay, and obviously the percentage, the efficiency of drug A will be comparatively greater than drug B. So every time the drug administration is being studied, we need to understand that the rate theory, the effectiveness of a drug with respect to time should also be considered. Okay. The next is induced fit theory. We need to understand that drug will bind at the receptor site, and drug is actually supposed to be two kinds. I'll show you in the next diagram. Okay, so induced fit is, we need to have a drug molecule that should best fit at the receptor site. You can easily see here in the diagram, this is a drug molecule. Okay, the molecule that we are considering to be the ligand molecule or drug molecule, and the structure is such as that it is easily binding to the receptor site. Okay, here you can see it is binding to the receptor site and it is starting its action, the drug action or the effectiveness. Okay, so this is said to be the induced fit theory, where we are considering drug molecule is binding to the receptor site. It is best fitted to the receptor site and it is showing its action, binding and activation. Okay, so affinity is being studied and we need to understand the efficacy or the response of the drug will totally depend on the moment the drug will bind and it will be activated. Okay, so here you can see in the diagram there are two kinds of drugs. There is agonist drug and antagonist drug. Agonist drug is a drug that is binding at the receptor site. It is having an interaction with the receptor site, okay, as well as it is starting its action. The response has started. Okay, so we are going to study about the affinity as well as the efficacy of the drug. The the agonist drug, is having a defined affinity and it is having a defined efficacy as well. On the other hand, there are certain drugs that are supposed to be antagonist drugs. So what these antagonist drugs are doing, they are going and they are fitting at the receptor site, but what they are doing is they are not leading to any kind of response. No drug action is starting and there is no effect. Okay, so the drug is not helping for the treatment of disease. So such kind of drugs, which is having a defined chemical structure, suitable chemical structure, and we know that it is having all the properties for the treatment of the disease, but this is not helping for the treatment of the disease because it is not starting its action, not starting the effectiveness. Okay, and this can be just because it can be a possibility ki any neurochemical or any hormone, okay, it is just blocking its function. It can be a possibility that the drug molecule, the antagonist drug, is binding here but its action is not being activated just because another molecule had binded to it. Or it can be another possibility that instead of this antagonist drug molecule, some other molecule must have binded at the receptor site. So the molecule is not reaching at its site of action. We have just discovered that this is a suitable receptor site. Probably our drug will go and bind to the receptor site. But what is happening? The site is already covered by some other molecule. The molecule is not going and binded to this particular receptor site. So this can also be a case where we consider the drug is not showing its action. Okay, so this should be considered when we go with the study of our drug discovery and development. Okay, so as I discussed earlier that when we are going with the development of the drug molecule, we need to have an idea or a hypothesis that what kind of a drug, what kind of a disease we want to go for. Okay, and based on the idea and hypothesis, we will move forward with the development of the drug, with the designing and synthesis of that particular compound which we consider to be a suitable compound that will help us to the treatment of disease. Okay, then we will go with the study or the effect of this compound on the tissues, on the animals, where we will study the preclinical trial phases, and when we'll go with the clinical trial phases, then we'll study the effect of these drug molecules on humans. Okay, if we get good results, then only we can claim them to go for the process of approval. In the drug federation, we need to send our drug. So telling them this is a good, showing a good therapeutic action, and actually it can be led to the market to be used by the consumers. Okay, so this can be done with the whole process of drug development. This is a general plan. When the pharmaceutical companies, they go, then they first thing they do is they choose a disease, they choose a target for their drug molecule, then they keep finding out a lead compound. A lead compound is a molecule where we suppose this will work for the treatment of the disease. Okay, and then we need to optimize the lead compound, optimize the structure, a suitable structure that will bind to the receptor site. Go with all the clinical trial phases, preclinical or clinical trial 1, 2, 3, 4. Okay, and then the market, the drug will be available in the market. So if we are considering a novel drug, a new chemical entity, then we need to understand that the pharmaceutical companies, they are taking a minimum time period of 10 years to develop a new drug. And obviously the amount of money is being invested is also too much. It is around $500 million or more than that. Okay, and when they are testing the compounds, it is around these number of compounds. And what for? Just for a single drug. See, developing or just production of a drug molecule is something different. They have discovered the drug molecule and they are going with the production of the drug molecule. That is a different task. And going with the production or development of a new drug, that is a different task where it will take a minimum time period of 10 years. Okay, because going with the trial phases, testing the effectiveness of the drug, it takes a lot of time. And if side effects are there, then we need to move ahead and just modify the drug and just go on for more trial phases. Okay, so first thing, choose a drug. Whenever we want to go for the drug development, choose a drug. For that drug only, we can just go for the designing thing. Okay, so when we have chosen a drug, choose a drug target, the place where your drug will go and bind. Okay, after you have chosen the target place, so you want to have a bioassay, a test where you can just determine the biological activity of the compound or the molecule or the drug or the ligand molecule, which we suppose will go and bind at the target site. So we need to have a bioassay that will be suitable both for when we are going for our in vitro testing as well as for in vivo testing. Okay, then we need to find a lead compound. Okay, so I'm using actually different words. I'm using this medicine, drug, lead compound, ligand. Okay, so when I'm talking about, see, in the common language, we call it as a medicine. Okay, in terms of bioinformatics, pharmacology, we call it as a drug molecule. Okay, so I'm taking the name of drug. When I'm taking the term called lead compound, so I'm talking about that suitable compound which I'm supposing is having all the suitable properties and which will definitely work as a suitable drug molecule. Okay, so that is said to be a lead compound. Then when I am taking the term called ligand molecule, then I am talking about the virtual process, the process of computer-aided drug design. I am considering that this is a ligand, this is a structure which I am creating virtually. Okay, I am designing using the in silico techniques, and I'm trying to understand, is it complementary to my receptor site? So that is supposed to be the term ligand form. Okay, so lead compound is actually the structure that is having some activity against the chosen target, but that is not having any good information about the drug itself. Okay, so we need to understand whenever we are selecting the lead compound, we need to have certain properties for the lead compound, and based on those properties, we will keep on selecting the lead compound to just go for the selection of the most optimized lead compound that will be the drug in future. Okay, so while selecting the lead compound, we can also go with the synthesis of analogs of that lead compound. Analogs as in, we can go for number of the complementary structures for a lead compound. Okay, and the moment we have gone with the creation of the lead compound, we are just okay that this is the defined structure. Then what we will do, we need to understand that what is the relationship of the structure and its activity that is being studied under the structure activity relationship model. That includes two models, the quantitative structure activity relationship model, and the other one is quantitative structure property relationship model, QSAR and QSPR. Okay, so how to select a lead compound? I told you lead compounds are those compounds which are actually helping us to go for the selection of those compounds which will probably go and help us to the treatment of the disease. Okay, so here you can see the lead compound discovery and optimization is a long process where we need to keep on discovering the lead compound. We can go, if it is a protein structure, we can go, or any other molecule, we need to go with the study of that three-dimensional structure. We can modify and model and design the structure totally based on if it is there suitably binded at the receptor site. Okay, and based on this, we can keep on optimizing the structure of a lead compound. But how to select the lead compound? the lead compound which is actually being selected, we can just select them by all these methods. We can go for compound selection based on serendipity, possibly certain accidental discoveries had been occurred, and based on that these lead compounds can be discovered that this molecule is actually working for the disease that are targeting the liver or targeting the kidney or such different areas. Okay, then from natural resources also. Okay, so we can pick the compounds from all the natural resources, and we can go with the optimization of the structure of a lead compound. Okay, then there are certain substrates, inhibitors, and what about the different mechanism of actions which are going inside our own body. So we can select any kind of active compound from that process as well. Okay, and we can go for certain other diseases as well, similar to the X disease for which we are going to create the drug. We can go for similar diseases also, and we can select an effective drug, and we can go for the creation of a lead compound selecting that particular drug molecule. Large scale screening means we are selecting the compounds from a big chemical library. We are trying to refine all those compounds which we consider are having suitable properties and can work for the treatment of a disease. Okay, or for the side effects of certain existing drugs, we can also consider the side effects, and we can find out that there are certain drugs which are helpful for the side effects. There is one drug that was actually being minidoxil is the name, and it was actually being discovered for hypertension, but later on it was helpful for the hair growth problems. Okay, so such kind of drugs can also be used. Experimental screening also helps. Okay, so we can go with screening, trying to search out that the drug is helpful for the treatment. Experimental screening will help us selecting the drugs, checking the action of each compound. Okay, and the computer screening obviously, because selecting all the properties, virtually going with the computational study of the properties and studying how it will go and bind to the receptor. So all this will help us to just go for the selection of a lead compound. Okay, so going with the selection of the lead compound, checking the structure activity relationship, then we need to identify the pharmacophore. What about the structural features that are directly responsible for the activity? And we need to optimize the structure. We need to understand that the features should be suitable enough, should be suitable to just produce an activity that is okay enough and help a drug molecule to just get and interact with the target molecule and improve the property. Okay, so with all this, we will just go with the determination of the toxicity and the efficacy of such molecules, such compounds in animal models, with the determination of the pharmacodynamics and pharmacokinetics properties. So pharmacodynamics, obviously I told you, it is dealing with the property that how the drug is acting on the body and how the body is reacting towards that drug, that is being explored under pharmacokinetics. Okay, so pharmacokinetics, it includes the study of absorption, distribution, metabolism, excretion, and toxicity, how the drug is being absorbed and how is it distributed across the body, what kind of effect it is showing in the metabolism of each and every cell of our body, and what percentage of drug is being excreted out. Because when we are taking it, the bioavailability of the drug is very much important, that how much amount of drug is administered at the site of action. Okay, it shouldn't be a percentage that is good enough, that is helpful for the treatment of the disease. It shouldn't be that a major portion of of the drug is being excreted out from the body. Okay, and lastly is we need to understand that whenever we are consuming any compound for the disease, treatment of X disease, we shouldn't have any toxic effects that are causing any other Y disease. Okay, and this Lipinski rule of five. So whenever we are going with the properties, with the study of a probable drug, a molecule that is suitable for just being converted into a drug molecule, we consider these rules said to be the rule of five. Okay, molecular weight considering. So this rule is basically considered just because we need to understand the molecule is traveling from one cell to the other cell. So it should have a molecular weight of less than 500 Dalton. The log P ratio should be less than 5. Hydrogen bond donor and acceptor should be less than 5 and less than 10. Because all these properties, they should be there for a compound, because then only the compound will easily go and travel across the cell membrane and move from one cell to the other cell. Okay, so after this, then all the processes we have just gone with the creation of the drug. See, with the determination of the properties and creation of the drug. So the drug creation is based on when we are going with the computer aided drug designing. First, we will go with the virtual designing of the drug. So what about computer aided drug designing? I'll talk about that in my next session, because I'll talk about the two major types of computer aided drug designing, where we'll be just discussing about how the drug and receptor binds to each other. What about ligand based drug designing, and what about the structure based drug design? If all this is okay enough, okay, the structure is okay, the ligand structure and receptor structure, all the properties, starting with the pharmacodynamic kinetic properties, structure activity relationship, if we have done with all this, we see that okay, all these things are okay enough. Okay, then we need to go for patenting of the drug. The first thing is we will go for the patenting of the drug, and then we will go with the study of the metabolism of the drug inside the organism, for which the diseases, for which the drug molecule is actually being developed. Okay, for example, for humans, we will check what effect it is causing on the basic metabolism of the cells. And obviously, we will continue to test for toxicity as well. So how we will go for the toxicity check, I'll show you in the next slide. And then we need to go for the designing and manufacturing of the whole process. Okay. And later on, we will go for clinical trials. We need to go for preclinical trials with animal testing and clinical trials with patients and healthy volunteers. And the moment we find that the results are good enough, then we can go to let the drug be market in the be there in the market with the approval of the drug federation. Okay, so if I'm talking about the toxicity study, so all this should be studied when we are checking with the toxicity. If we need to undergo that the drug is not causing any tumor induction, carcinogenicity, then teratogenicity. If it is causing any kind of development abnormalities, we need to understand this also. If it is causing any kind of mutation, okay, so mutagenicity is also being studied. Local toxicity like the dermal, ocular, inhalation, vaginal, and rectal, all these toxicity are also to be considered, as well as the effect on the reproductive performance. Okay, so while going with the development of the drug and with the study of the metabolism of the drug on the organism, we need to study the effects of the toxic effects of the drug on the body, including all these special kinds of toxicity. Okay, so with the clinical trials, with the trials, see, with the preclinical trials, with the animal testing, you need to just check what is the effect of the drug, how the drug is being administered, how is it working in the body of your animal. Okay, so mostly they take rodents and they go with the study of how they are just responding to the drug. Okay, and then we go with the clinical trials. So in the clinical trial phases, I told you, healthy volunteers, they are actually working. So when the drug is being tested on healthy volunteers, so the reason is they want to understand what is the toxicity effect. Is there the dose, the amount of dose which is being taken by the healthy volunteers? So what kind of toxic effect it is causing? Is it causing, and what percentage it is causing? Okay, and based on this, they are just screening up what about the side effects, if there are any unexpected side effects which they have not thought earlier while the development of the drug. So all this is studied in the phase 1 of the clinical trials. Okay, in the second phase, phase 2, we come up with we need to study about the drug, how we can just test on a small group of patients, and we need to understand that is it causing any kind of beneficial effect. If it is helping for the treatment of the disease, then we need to understand what amount of dose is actually working for this effectiveness. Okay, and what if we increase the dosage? Okay, so what kind of effects the drug will lead to? And in the next level, the phase 3 will be when we will talk about a large group of patients, and we need to understand what about the treatment of the disease. Okay, last phase comes to the phase number 4, where we consider, okay, the the things which we have gone, all the phases, we have just crossed all the phases. We understand that the drug is okay enough, it is helping very much in the treatment of the disease, and the pharmaceutical companies, it is like now it should be there in the market for the use of the patients, for the consumers in public. Okay, so the moment it is there in the market, obviously after the approval of the drug federation, then also the monitoring of the side effects is keep going. Okay, if any kind of side effect is there, then obviously the drug will be banned from the market, and it will go back to the pharmaceutical companies to just go with the modification and check what kind of side effects and how we can go with the removal of the side effects. If it is okay enough, then the medicine is being consumed and be there in the market. Okay, so all this was there for the development, and the role of computer aided drug designing will be where we will talk about how the drug, the ligand molecule, it binds to the receptor, what is the interaction, and what about the process of computer aided drug designing, talking about structure based and ligand based. Okay, so this is the end of my today's session, and I will just take the questions now.